Welcome to this explainer. Today, we're going to approach virology exactly like a master detective profiling a shape-shifting suspect. We are decoding the foundational rules of how viruses actually operate, how they mutate, and how they spread across the globe. As you'll see, a virus isn't just some random microscopic bug. It's an incredibly intricate evolutionary machine. Here is our roadmap for today. We'll be hitting 5 key areas: classifying the viral world, the Baltimore system, engines of viral evolution, the quasi-species cloud, and finally, tracking viral outbreaks. Let's get right into it. Section 1. Classifying the viral world. Identifying the suspect. To begin profiling our suspect, we run into a massive biological hurdle right out of the gate. Think about it. How do you classify something that doesn't even fit into the standard 3 domains of cellular life? Because viruses aren't technically cells, traditional biological sorting completely falls apart. So virologists had to stop just looking at a virus's physical shape under a microscope and start analyzing its genome architecture and its underlying replication strategy. To bring some order to this chaos, the International Committee on Taxonomy of Viruses, the ICTV, established a really strict hierarchy. It ranges from realm at the very top all the way down to species, using standardized suffixes like viralis for an order or viridae for a family. And the genius of this system, it ensures that scientific literature across decades and across laboratories remains perfectly comparable. By using a formal species name, diagnostic labs can anchor highly unstable, constantly mutating clinical strains to one rock-solid taxonomic record. But here is where it gets absolutely wild. Modern technology has totally flipped taxonomy on its head through sequence-only taxonomy. Thanks to metagenomic sequencing of soil, ocean water, and human microbiomes, we're discovering an absolute flood of viral sequences. This has pushed the field to name and classify viruses based purely on genetic signals, without ever growing or culturing them in a lab. Now, as you can imagine, this has been a highly contested shift from the old days of microscopy, because it commits the field to naming entities that we might literally never physically see. Okay, Section 2, the Baltimore system, how the suspect operates. Moving past basic identity, we desperately need to know how our suspect actually operates once it gets inside the host. Well, to understand viral operations, you have to understand the ultimate viral goal, messenger RNA, or mRNA. This is the essential genetic transcript that every single virus in existence must figure out how to produce. Why? Because mRNA is the only language the host cell's ribosomes actually understand. To force a cell to manufacture viral proteins, the virus absolutely must generate mRNA. So back in 1971, a scientist named David Baltimore decided to ignore physical morphology entirely. Instead, he asked one unifying biochemical question: what specific pathway does a virus use to generate this messenger RNA? And the results of that question gave us the 7 Baltimore classes. Literally every known virus sorts into one of these 7 routes. For instance, Class I viruses use double-stranded DNA and just mimic normal cellular transcription. Class 4 viruses carry positive-sense single-stranded RNA that is directly infectious. It can act as mRNA the very second it enters the cell. And then you have the rule breakers, like Class VI retroviruses, which start with RNA but use an enzyme called reverse transcriptase to create a DNA intermediate. Now, this isn't just academic trivia. It's intensely practical. For a diagnostic scientist, knowing a virus's Baltimore class is like having its operational playbook handed to you. It immediately predicts crucial clinical details. It tells you if reverse transcription has to precede amplification during lab testing, like we see with HIV or hepatitis B. It tells you exactly where in the host cell the virus replicates, whether that's the nucleus or the cytoplasm, and it instantly highlights which viral enzymes we can target with antiviral drugs. Section 3. Engines of viral evolution. Changing disguises. Once we know how they replicate, we can start to understand their ultimate weapon, their astonishing ability to change disguises and evolve at lightning speed. To really grasp this, consider the mathematical chasm between the mutation rates of RNA and DNA viruses. RNA viruses typically mutate at a rate of 10 to the power of -3 substitutions per round of replication. DNA viruses, they're way down at 10 to the power of -8. That is a difference of several orders of magnitude. RNA viruses generate massive genetic diversity on a timescale of mere days, whereas DNA viruses remain remarkably stable. So why the huge gap? It basically comes down to the no proofreading rule. When our own cells, or DNA viruses, copy their genome, They have an exonuclease, a built-in spell checker that removes mistakes. Most RNA polymerases completely lack this proofreading mechanism. It's like typing an essay without a backspace key. Every mistake becomes permanent. Now, there is one notable exception. Coronaviruses actually possess a dedicated proofreading enzyme called NSP14. But for most RNA viruses, this constant stream of uncorrected errors results in rapid escape from our neutralizing antibodies and antiviral drugs. It's also exactly why diagnostic molecular assays for RNA pathogens require constant sequence surveillance and redesign, just so they don't lose sensitivity. But mutation is just one engine. Let's compare two specific evolutionary mechanisms side by side: antigenic drift versus antigenic shift. Drift is the slow, incremental accumulation of point mutations. It's driven by the pressure of our immune systems, and it's the exact reason why we have to reformulate the flu shot every single year. Antigenic shift, on the other hand, is a sudden, large-scale event. When a virus has a segmented genome and two different strains infect the exact same cell, they can freely swap whole genome segments. This reassortment creates a radically new virus with surface proteins humanity has literally never seen before, triggering massive global pandemics like the ones we saw in 1918 and 2009. Section 4. The quasi-species cloud. A gang of clones. Because of these rapid engines of evolution, we really have to rethink what an infection actually is. It isn't a single suspect, it's a quasi-species. A quasi-species is basically a cloud of closely related viral variants generated continuously by that high error rate we were just talking about. When a patient is infected with hepatitis C or HIV, they don't just harbor one uniform virus. They harbor an entire vast population of slightly different genome clones inside their body. Grounding this right at the clinical bedside, check out this powerful quote. Antiviral treatment failure is frequently explained by pre-existing minority variants within the quasi-species rather than by resistance arising entirely after treatment starts. This completely reframes how we view drug resistance. The resistant disguise didn't necessarily form because of the drug. No, it was already present in the cloud of variants at a very low frequency, just waiting for the selective pressure of the drug to wipe out the competition and make it dominant. Because of this quasi-species cloud, our diagnostic tools had to evolve too. We're seeing a major shift in the lab. We are moving away from simple consensus sequencing, which only shows us the dominant, most common variant in a sample, and we're moving toward deep sequencing. Modern scientists have to use deep sequencing to spot those dangerous minority variants hiding at levels as low as 1% of the total population. You have to find them before a patient even begins antiviral therapy, or the treatment might be completely doomed to fail. Section 5, Tracking Viral Outbreaks, the Final Pursuit. So how do we take all this knowledge about genetic diversity, mutation, and taxonomy and actually use it to hunt down the suspect during a live global outbreak? The answer is molecular epidemiology. This is the practice of applying all that viral genetic diversity we've been discussing to answer incredibly practical questions, like who infected whom and where exactly did the outbreak start? Every single mutation and every reassortment leaves behind genetic breadcrumbs. By analyzing these genetic changes, scientists can reconstruct evolutionary relationships on a phylogenetic tree, pinpointing the exact geographic origin of an introduction and estimating how long a virus was circulating before we even noticed it. Let's look at how this builds dynamically during a crisis, like what we saw with Ebola in West Africa or the global spread of SARS-CoV-2. A virus emerges in a matter of days. Within weeks, real-time sequencing is happening. Phylogenetic trees are built shortly after, directly informing the ongoing public health response. This means local scientists on the ground, even in resource-limited settings like Bales Estate, can interpret transmission chains in real time, flag changes in transmissibility, and directly inform how they manage patients right there on the floor. As we wrap up this explainer, I want to leave you with a somewhat provocative thought. If we can now identify, classify, and track viruses we've never cultured or even seen using sequence-only taxonomy, how will this invisible detective work change our readiness for the next pandemic, the next disease X? As metagenomics continues to map an entirely invisible world of pathogens, are we finally gaining the tools to stay one step ahead of biology's ultimate shapeshifters?